Hey guys. Okay, so for some reason my video lecture did not want to go through for this one. I don't know why. There we go. So I'll just do it again really quick. Okay, so this last lesson we have is on DNA, RNA, and proteins. Okay, so first thing we're going to talk about is the central dogma. Okay, the geneticists accept that the basic mechanism for reading and expressing genes is from DNA to RNA to protein. So what we're going to do is we're going to go from DNA molecule to an RNA molecule, and then we're going to do is an RNA molecule to a protein molecule. Okay, we have two different processes. First is transcription, and the second is translation. So in the easiest, simplest way to explain these two processes, transcription and translation, in your own words, hint, hint, is transcription is going from DNA to RNA, and translation is going from RNA to a protein. Hint, hint. So if you watch this video, you'll have a good idea what I mean there. Okay? This is referred to as a central dogma of biology. DNA codes for RNA, which guides the synthesis of proteins. Now, why is that even important? Well, people talk about all the time, protein, 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 proteins. Proteins make up they're so essential. Okay, they're essential for your health. They're essential for understanding certain cells, what their structure is, what their function is, and if certain cells have specific functions, then that's going to turn into different functions for that organ and organ system, and so on and so forth. Proteins have such a high importance in the world of biology. Okay, so before we get into this transcription, we got to know what RNA is and also if there's different types. Okay, RNA is a nucleic acid similar to DNA. Okay, but with the sugar ribose. Okay, and with uracil instead of thymine. So if you are working with RNA, remember, in DNA, adenine and thymine matched up. Okay, so adenine and thymine, and cytosine and guanine. Now, in RNA, cytosine and guanine are still together. However, we have thymine is no longer. Okay, thymine is no longer. We actually have A, an adenine, and U in uracil. So A and U match up, and I'll show you in a example below here. There are three major types of RNA, and they all have specific jobs. First is messenger RNA, and that's the main part of RNA that we're going to be using or turning into in transcription. We have ribosomal RNA, hence rRNA, okay? And then we have transfer RNA, which is tRNA. Okay, so we have three different kinds: mRNA, rRNA, and tRNA. Okay, here's a good little example of what they look like. Okay, so you're going to see mRNA, messenger. I like to put it like the Morse code. You have dashes with Morse code doesn't have colors, but you have different coding, right? At messenger RNA, you're sending a message. Okay, ribosomal or RNA, rRNA is kind of like a glob, and it's essentially they help form ribosomes, right? And in the ribosome, or in the cell, a ribosome is like a circle, so that's the easiest way to recognize that one. And then tRNA, or transport RNAs, or transfer RNAs, excuse me, look a little bit different, and they have a specific function that we're not going to get into. That's more molecular biology. Okay, so mRNA it carries genetic information from DNA in the nucleus to direct protein synthesis in the cytoplasm. Right, they're carrying their messengers. Okay, rRNA is associating with the protein to form the ribosome. Again, R ribosome, and the transport amino acids to the ribosome is transfer RNA, tRNA. So they're transporting amino acids. That's exactly why they're called what they're called. Okay, and then that moves us into transcription. Okay, the first step of the central dogma involves the synthesis of mRNA from DNA. Boom, there it says it again, from DNA to mRNA. Okay, in a process called transcription. The easiest way to explain it in your own words. Okay, the enzyme RNA polymerase regulates RNA synthesis. Okay, so there's a polymerase, there's a specific enzyme that regulates this synthesis. Now, RNA polymerase moves along the DNA strand in a 3' to 5' direction. Okay, so they're going to synthesize that mRNA specifically because of the direction it's going. So RNA is transcribed in the 5' to 3' direction. So what you're going to get is this unzipping Okay, so if it goes from 3' to 5', it's taking that part off, so it's unzipping that DNA strand, and we're making this copy. And then what's going to happen is RNA is transcribed into 5' to 3 direction. Remember, it has to go in the opposite way. Okay. RNA processing. In comparing DNA code with this transcribed mRNA, scientists found that the mRNA code was much shorter. So they just found out that the mRNA was shorter than the original DNA strand. DNA sequences not found in the mRNA are called introns. Okay, so they found that there were actually some segments or parse sequences that were not found. Okay, and the DNA sequences that remain in the final mRNA are called exons. Okay, so they found out why this was shorter is actually because we no longer had some of those DNA sequences. Oh, the code. Okay, the scientists hypothesized that the instructions from protein synthesis were encoded in DNA. Experiments during the 1960s demonstrated that the DNA code was a 3-base code. Okay, DNA and amino acids are all, or amino acids, not DNA, amino acids are all based off a 3 code. Okay, 3-base code in DNA or mRNA is called a codon. Okay, so when we're talking about DNA and mRNA, the amino acid sequence is a codon. So this circular, I love this diagram. How most of us were taught was in a box, and it was kind of confusing, but this is just, I love this. So what you're going to do is, this is a dictionary of the genetic code. Okay, notice that all but 3 codons are specific for any amino acid. So these 3 are stop codons. So what you'll see is on the outside here, okay, are different amino acids. So we have things like alanine, arginine, proline, glutamine, histidine, lysine, so on and so forth. So this shows their coding. So you always start in the middle, always. So A, C, U, or G. Okay, so now you know we are working with mRNA. Why? Because we have U and not T. So if you have something that, say the amino acid code is UGG. So you go from U to G, and you see that that specific amino acid is tryptophan. Okay, say we have CUA. We go C, then the next letter is U, because the next one comes in the blue, so CU, and then the last one comes on the way outside with the small ones. Okay, CUA is leucine. Okay, it is going to be very, very, very important for you to understand how to do this process, and will benefit you in the future. Okay, we talked about transcription, so now we got to talk about translation. So after synthesis, we go from mRNA, or RNA, very simply, to an amino acid. Okay, or protein, however you want to put it. Okay, after synthesis, mRNA moves from the nucleus into the cytoplasm, where it connects at the 5' end to a ribosome. Okay, remember, so we went from 5 to 3, so now it's got a hook on that tail end to another 5', because it has to be opposite. So the mRNA code is read and translated into a protein through a process called translation. So what it does is it takes that mRNA, that messenger RNA, and it hooks onto a 5', and now it translates it into a protein. Okay, it translates it into a protein, which now can be used at the cell's disposal, however it needs. Okay, translation tRNA molecules act as the interpreters of the mRNA codon sequence. The tRNA is activated by an enzyme that attaches a specific amino acid to the 3' end. So that, remember, that code is going to determine specifically what amino acid that is with that circular dial that we looked at. So the middle of the folded tRNA contains an anticodon, a complementary sequence to the mRNA. That's not exactly the most important thing that I want you guys to know. I put it on there because it's a part of this chapter that they want to highlight, but I'm not super fond of how they teach it. And then lastly, the role of the ribosome. Okay, ribosomes provide a site for protein synthesis. Again, protein synthesis is huge. When mRNA leaves the nucleus, the 2 ribosomal subunits come together to hold the mRNA in place for translation. So they're going to hold it in place. Why is that important? Well, if something's going to be copied, you don't want to be moving all over the place. You want it to be still while you're doing it. The ribosome structure has grooves that serve as tRNA sites for amino acid attachment. You want that amino acid to be attached, so therefore you can have better transportation of, or not transportation, but transfer knowledge of what you're wanting that specific amino acid to do. Okay, Now, I just wrapped up a long lecture in about 9 minutes and 55 seconds, okay? I understand if this may be a little bit confusing to you. Understand this process, how to figure out an amino acid. That is huge. That's one of the biggest things I want you to come away with, okay? The second and third thing I want you to come away with is, second, what are the 3 different kinds of RNA? Is there different ones? Yes, there is, okay? If we are RNA, what does that mean? We actually have uracil. We do not have thymine, right? And then the last thing, probably the most important thing is, please, please, please understand that transcription is going from DNA to RNA, and translation is going from RNA to a protein. Hope you guys are all doing well. See you guys.